Hello. Immunity. It's complicated. It's really, really complicated. All the other systems could be kind of figured out by hand. You could figure it out maybe with a microscope. You could do experiments. You can apply basic physics, mechanics, fluid mechanics, um, physics of heat, physics of, movement, those kinds of things could be applied to figure out how heart works, how lungs work, those kinds of things were relatively easy. Um, Ancient Greeks dissected dead people and monkeys and all sorts of other animals and figured a lot about our bodies, things that they could see. They got a lot wrong too. And then for the next, I don't know, almost 2,000 years, nobody dissected a human being. Nobody dissected anything. Everything was written by ancient Greeks, and that those were medical books, textbooks survived for all these centuries, and nobody was making any new medical progress because most of the medical progress was based on armchair philosophizing until about late 16th century, where some people like Vesalius started dissecting human beings, and late 16th century uh, into 17th when the first microscopes were made, first by Jensen and then by Robert Hooke. But those microscopes were primitive. They could enlarge but not really increase the visibility. They didn't have the ability to see much more. And they put all sorts of plant materials and so protists in the water. There's all sorts of things, and of course some of the bodily fluids were the first things to be viewed under the microscope. Sure you all had biology classes where you did took a little toothpick and scraped a little bit of the inside of your mouth and cheek and looked at under the microscope and saw all sorts of kind of bacteria and stuff and cells and whatnot under that. So that was one of the first thing that was looked under the microscope. Interestingly, another one of the first things that was looked at in a microscope was sperm. Hard to see because those cells are really tiny. And then, of course, blood. And blood has a lot of interesting cells in there, and the early microscopists uh, pretty quickly figured out how to classify them. So you had all these, neutrophils, basophils, and whatnot, monocytes, and so with that knowledge for a long time, for the next couple of centuries, um, that was all we could know about immunity. We kind of figured out that those white blood cells have something to do with the defense, because you see a whole bunch of white blood cells fill in the blood when a person is sick, and different types of white cells with different types of sicknesses. So it took a while to figure out, the existence of bacteria, existence of viruses, existence of protists like malaria, parasites, and of course intestinal worms, those kinds of things. And so depending on kind of what kind of disease the patient had, they could see the different types of white blood cells were present in the blood in large quantities. They could see that some of the cells were attacking, let's say there are thousands of them attached to a worm, a parasitic worm, and killing it. uh, so that's pretty much what they could do. And they figured out vaccination from some other ideas. They really had no idea how it works. They just realized, through experimentation, that it works. So they got the smallpox vaccine, uh, and then some other vaccines later on, uh, using kind of the logic and using experimentation, seeing what works in patients without understanding the underlying mechanisms. None of that could be done until at least 1960s. because that's when we figured out that DNA is molecule of heredity, and that also molecule that stores information about proteins that are used by all the cells for all the functions. Until then, we couldn't do anything about immune system. We could study the physics of the mechanics of the heart and mechanics of the lungs, but immune system was opaque to us. So discovery of the structure of DNA has allowed people to start developing better and better laboratory techniques for studying genes, DNA, and also their products, proteins, and sugars that are attached to those proteins, and understanding the cell better and better. It took a few decades to refine those techniques. So right now, immunology is really the only one system that you, at this level of anatomy and physiology, need to study the molecular and cellular level because that's where it operates. Um all the other systems you can understand at a kind of an organ level. Immune system makes sense only on the cellular and molecular level, and this makes it really, really difficult. Another thing that makes it difficult is that the knowledge about immunology is exploding. What you read in your textbook about immunology is bare bone basics, and it's so difficult, and there's so much more, and there's so many immunology papers being published every day. Just reading the literature would be a full-time job, so nobody does it except immunologists, which means people who are not immunologists have usually a little outdated and basic understanding of immunology. That includes me. I'm not an immunologist. That includes a lot of people writing about immunology right now because of COVID who are not immunologists. So if you see an article that's written by a dentist or ophthalmologist or urologist or cardiologist, be very careful. Try to just read things that were written by immunologists, virologists, and epidemiologists because those are very, very complex uh, systems and very, very complex areas of study, and people who are not specialists usually don't know well and they don't know enough. And that includes me. On all the other systems that we study, uh, it's relatively easy for me to learn here and there more, often much more than there is in the book. But immune system, I'm pretty much at the level of the book. I don't have the time or even ability to understand all the research that is currently coming out. So that makes it very, very difficult. It's all molecular cellular, nothing you can hold in your hands, nothing you can understand by intuition. And it is so complicated. And yet it's very relevant. We are in the middle of the epidemic right now, and it's very relevant to understand how immune system works. So at least get the basics. So when you read some of those articles, you can figure out, okay, this is a hoax, or yeah, this sounds right. Um, when you go to subfolder topical studies and open that document about how to do topical studies, there's a list of sources on the bottom. And those are trusted sources because although articles written uh, in those places are mostly written by science writers, science journalists, they're not your regular journalist who covers crime and school boards and stuff like that. Those are people who have background and education in science and have spent some years studying the details of science. They know how to interview scientists, they know how to read scientific papers, so they can be trusted. They also work for media organizations that are making sure that their articles are accurate, that they're scientifically correct. So if you go to New York Times Science, or if you go to Eon, or if you go to Scientific American, their coverage of viruses or immunology or epidemiology is likely to be correct, even though the author is a journalist and not an immunologist. That journalist has learned a lot and knows how to find the most relevant, the best immunologists to interview to get all the information and making sure that that article is correct. So if you find just somebody posting something on Medium or Facebook, don't trust that, but you can go to trusted sources to learn about immunology. So back to the basics. What do you need to know about immunology? Well, the most basic things is that there are three layers or three levels or three degrees of defenses. First are barrier defenses. Um, Those are all the methods that the body uses to prevent foreign organisms, particles, and stuff like that from entering the organism in the first place. The most obvious is skin. You studied skin last year. You know it's thick. It has many, many layers. It has some, macrophages right in there, and it's really hard for any kind of foreign organism to just penetrate the skin and go inside. So it's a very, very good first uh, line of defense to have skin. Of course, the skin has to be healthy and intact. If you have uh, an injury, a cut, that's an entry point for bacteria, viruses, all sorts of other things. There are places in the body that are not covered with skin because our inside of our body needs to communicate with the outside of our body. So there are all sorts of openings: the eyes, the nose, the mouth, anus, all sorts of other places where um, where it's necessary to come up with different kinds of defenses. Those are still called barrier defenses. So, for instance, there are several places, several glands that, or types of glands that secrete uh, antibodies, immunoglobulins, usually A, uh, that are found in the saliva. So, as a first defense in the mouth when we eat something. Uh, and some of the types of sweat glands, again, that was last semester, some of the urinary secretions, some of the vaginal secretions. So those are kinds of places where antibodies are most important to be there because they are um, going to, grab whatever is coming into those orifices, into those openings. Respiratory system has its own problem that air is just flowing freely in and out of the lungs. So nasal passages and as so upper respiratory tracts as well as the lower respiratory tracts have all sorts of tricks. They are secreting mucus. That mucus may also have some immunoglobulins, uh, and they have cilia. They have pseudostratified ciliated epithelium, which has cilia that are constantly beating, and they're beating towards the throat. So cilia in the nose are beating down towards the throat. The cilia in the trachea and bronchi are beating upwards, pushing all the gunk and goo and dust and pollen and bacteria, whatever got into the trachea and bronchi, pushing them up towards the throat. Once it's in the throat, you can deal with it. You can spit it. You can swallow it. Uh, there are ways to get rid of it. If you swallow it, what is going to happen? Digestive system also has its own defense. The first most important defense of the digestive system is that the stomach content is extremely acidic with hydrochloric acid, and almost nothing can survive that. Yeah, there are some bugs that can, and that can make us sick. But a lot of stuff, most of the bacteria and stuff like that goes into the stomach, gets uh, destroyed by the acid. So that's another barrier defense. So those are the barrier defenses that are either mechanically or chemically or in you know, using either physics or chemistry, uh, protecting us from the bugs even getting into the system, getting into circulation, getting inside of cells. There are macrophages also inside of alveoli. So there are various ways to protect us. Those are barrier defenses, the first line of defense. And then uh, after that, if the bug goes in there, there is going to be another layer of this defense, and that is called innate immune response. And those are cells that can recognize foreign bodies, at least some of them, and eat them. Eating by a cell is called phagocytosis. So those are neutrophils, macrophages, and monocytes. And then there are natural killer cells, which recognize when a cell of your body is infected, let's say by a virus, and then gives it a signal to kill itself. The programmed cell death is called apoptosis. So that will be the second line of defense. Now, those uh, types of defenses, phagocytosis or NK cells inducing apoptosis, they're very non-specific. They're going to recognize this is foreign. That's about it. Let's attack it. And they may or may not be very efficient. Uh, so then we have the third and most important layer of defense, and that is the adaptive immune response. Adaptive immune response involves both B and T lymphocytes, B and T cells. Both of those produce antibodies, and both of those produce antibodies that are very, very specifically binding to a particular pathogen. So they often have to learn. That's not learning the way our brains learn. They have to learn which pathogen is there and then make more antibodies against it. The difference between B and T cells is that B cells make a lot of antibodies and then shed those antibodies, release them freely into the bloodstream or lymph stream in order to for those antibodies to attach themselves to the foreign body and induce various killer cells to come and kill those organisms. T cells do not shed their antibodies; they keep their antibodies on the surface, and so when that antibody attaches to a foreign organism, the T cell is bound to that organism, which is kind of powerful because a T cell can do all sorts of things. It can recruit other cells to come and kill and swallow that foreign particle, foreign organism, bacterium, or whatever it is. So the B cells and T cells are going to make antibodies. And so your book explains in a decent amount of detail how do they make antibodies. So they make millions of different combinations of amino acids that make an antibody. And you don't have a gene for each antibody. You have, genes for, enzymes that are going to make 20 amino acids, the basic amino acids, essential ones, and those amino acids are going to be made, are going to be used for to make all those gazillions of combinations. You have 20 amino acids, you can combine them in many, many ways. So you can have really, really a large number of different types of antibodies, which are going to have slightly different polarity, slightly different shape, and thus can be more or less efficient at attaching themselves to different kinds of antigens on those are molecules on the surfaces of the foreign organisms and foreign bodies. So there are not, millions of genes for antibodies, uh, or otherwise our genomes would have to be much much larger than what they already are. So instead of having a genome that codes for every antibody, we have an immune system that can make every possible combination and see what works by trial and error. And some of those combinations are potentially dangerous in the sense that they can attack our own cells. And so those are mostly eliminated during the process of the development of the immune system. Again, your book gives a pretty good basic idea how those things happen, both for B cells and in T cells. How did they get rid of antibodies that are harmful to your own body, to the host? Once an organism, foreign organism, comes inside, uh, all these antibodies are going to be floating around, and some are going to be useless. Most of them are going to be useless, but there might be one or maybe two that are going to uh, be able to attach themselves to the foreign body and to induce the immune response, the killing. So recruiting, sending chemical signals, recruiting all sorts of cells that are going to come and kill the bacteria or kill the virus, the cell that contains the virus. And so those then go back to spleen, thymus, depends on the age, depends on, is it a B or T cell, bone marrow, and they are going to make many, many more of themselves. Now that takes time. Mitosis takes time, right? And so it takes time for those antibodies, either B cells making antibodies or T cells keeping antibodies on their surfaces, to make many, many more copies of themselves. In the meantime, those bacteria or viruses and whatnot are reproducing inside of your body. So it's kind of a race who's going to win. And often the, the bacterium or the virus wins and the person dies. However, if the person survives, there is a memory. There is a whole bunch of B cells and T cells that have encountered that organism and managed to defeat it. Slowly, hard, it was a long fight, but they managed to defeat it. And then, if that same organism comes in again, they can spring into action. They don't need to wait days for mitosis in order for them to have enough. They're going to be already there in large numbers and go. And while they're going into action, they are also reproducing more and more and more. So often, second time, you get uh, the same pathogen. Uh, your sickness is either very mild or you don't even notice. You don't know that you're sick because your immune system is working just fine. Um, so, as if you read a bunch of those articles that are posted in additional readings, you're going to hear a lot more about interleukins and about cytokine storms and those kinds of things. It's beyond our book, beyond our exams, but it's useful in order to understand what's going on with coronavirus right now because it's an unusual virus that does things differently, is very good at avoiding our immune system, and it might be T cells more effective than B cells. Kids tend to not suffer from COVID 19 as badly as adults, mostly because their primary, their innate system, their innate system is much stronger than in older people. So their very first encounter with just, NK cells and all that stuff, very non-specific. Hey, there's an intruder, kill. So no cytokine storms, and they can be pretty effective, which is why very few kids got sick and very even fewer kids died of COVID 19. So those are kind of things that I wanted to talk today. I don't want to go into greater detail here. What your book says is the most simple explanation of the immune system that exists. Those additional reading can uh, sometimes be clearer in explanation than the textbook. I have actually posted a couple of those links, not just in additional readings, but into the lecture readings in the immune folder, because I think they're especially clear. Uh, so you are going to hopefully understand it better, and understand how vaccines work using the primary and the secondary response of the adaptive system. So instead of getting sick and potentially dying from the first onslaught of the microorganism that attacked you, you inject either a dead virus or bacterium, or just an antigen, just a protein or sugar that's normally found on the surface of it, in order to stimulate production of large quantities of the antibodies. So when you do actually get infected by that microorganism, you don't have to go through the primary response anymore, because primary is risky, you may die. But you only get a secondary, which is much faster, much stronger, and has much greater chance of actually killing the intruder. So there's going to also be a topical study about explaining vaccines. So this is the time to kind of think about how you're going to write that one as well. All right, thank you.